Today we're going to be doing a bacterial transformation lab. Some of the materials we're using are E. coli, which is a bacteria, and we're going to be doing the transformation with a plasmid. This plasmid is called pGLO, and when you look at it, it doesn't look like much, it's just a clear liquid, but this plasmid actually contains two different genes in it that we're going to attempt to get our bacteria to actually incorporate this plasmid DNA into its own cells, and then it will hopefully be able to actually produce certain proteins that those genes code for, and we'll be able to see that when we plate them on different plates. Some background information for what I've already done is I've grown some E. coli, so this is called a starter plate. And so I've already grown this E. coli, and I'm going to use this as my starter E. coli colonies when I actually do my bacterial transformation. And I have made some plates that have different combinations of things in them in order to do this lab. So there is a plate that has just the regular, what's called luria broth. And so this is a regular plate. This is the same stuff that my starter plate was made on. And then I have two plates that have ampicillin mixed into them. And the ampicillin is what we would expect the bacteria to not be able to grow on, because ampicillin should kind of stop bacterial growth. My last one also has ampicillin in it, but it also has a sugar called arabinose sugar in it. And we'll talk about that as a class as to why this one is so different than the one that just has the ampicillin in it. So these are the plates I'll be using when I actually plate my bacteria, but I have to follow a series of steps in order to complete this lab before I put it onto the plates. So what I've already done is I have filled two tubes with what can be called transformation solution. This is calcium chloride. And when we do the pre-lab, we'll talk about the importance of this material and why it's being used. They're sitting on ice. For most of this lab, it'll be sitting on ice to kind of keep my materials cool and keep the cells and their membranes in place. But we will notice at one point we're actually going to put it in a hot water bath and heat shock it. And again, when we're in class together, we'll talk about the purpose for that. So if you're following along in the procedure that I've given you, I'm jumping in here at step number 4. I've already put the calcium chloride in the tubes and put the tubes on ice. And now I'm going to add the bacteria to it. So what I have here is a sterile loop, and I'm going to use this to pick up my E. coli colony. And I only want to pick up one colony when I do this. I don't want to really scrape from where you see these big streaks. I want to pick up just a single colony. I don't need that much bacteria to get this going. Now I'm going to add the E. coli to one of the tubes. I'm going to do this for both, so it really doesn't matter which one I start with. And when I put it in, remember this has the calcium chloride in it. I'm just going to kind of spin it around to make sure the bacteria actually comes off and ends up in the solution. I'm going to put it back in the ice, and I'm going to do the same thing for the second tube. Now I already have these tubes labeled. One of them is labeled plus pGLO, and one of them is labeled minus pGLO. So the minus pGLO, we won't be adding anything extra to it. We're just going to follow the basic procedure. The plus pGLO is the one that we will actually be adding the pGLO, the plasmid that I spoke about earlier, to in hopes of getting that bacteria that is in there to actually pick up that plasmid DNA. So I've added my E. coli to both of those tubes, and I'm leaving them on ice. Now I need to add that pGLO plasmid just to the one that says plus pGLO. The plasmid is in this tube like I showed you earlier. It just looks like a clear liquid. If you were to shine a UV light on it now, you really wouldn't notice anything. So it just looks like a clear liquid still. It's not glowing, nothing like that. This is important. We'll talk about that once we actually get our final results as to why it doesn't glow when it's in the tube, but why we do expect it to glow on one of our plates later. I'm going to open this up. I'm grabbing another one of my sterile loops. And when I put my sterile loop into the tube, I just want to make sure that I get kind of a covering on my loop, kind of a film covering the circle in the middle. That will be enough of the plasmid to make this work. So we don't need a ton. We don't need to like dump it in or anything. I need to make sure I find my correct tube. My purple one is the one I labeled as plus pGLO. So I'm going to open that up and do essentially the same thing I did with the bacteria. I'm just making sure that that material actually gets into the tube. All right. So I've done kind of my basic setup now. And what you'll see in your procedure is it says to let them sit on ice for 10 minutes before we move on to our next step. During that 10 minutes is when you would also label your plates. So I'm going to show you how I labeled them already, and we'll talk about why I labeled them the way I did. I guess I can cover up this E. coli. We don't really need it anymore. So if I look at my plates that are labeled, I see 2 of them say minus pGLO and 2 of them say plus pGLO. The ones that say minus pGLO are getting just regular E. coli on them. So they haven't gotten the plasmid DNA into them. And so what I would want to think about is, what do I expect to see on these plates? This plate right here, the one that just says LB, has nothing else mixed into it. It just has kind of the nutrients that the bacteria need to grow. So if I spread my bacteria all over this plate, I would expect a lot of bacteria to grow on that. So hopefully that's what we'll end up seeing. How is that different from the plate that has ampicillin on it? I'm going to put the same bacteria on there, the bacteria that doesn't have the pGLO plasmid in it, but this plate has ampicillin mixed into it. So I'd like you to think about is, do I expect to see any bacteria on there, or do I not? Now this is different from what I expect to see on these 2 plates that are labeled plus pGLO, because the bacteria that I'm putting on there, I'm assuming that if my bacterial transformation worked properly, has picked up that plasmid DNA that has the gene on it that allows it to actually grow in the presence of ampicillin. So normally ampicillin should stop the growth of the bacteria, but that plasmid that I've put into it has a gene that encodes for something that allows the bacteria to actually grow on the ampicillin. And if I've picked up that plasmid correctly, there's a second gene on there that, when it is grown in the presence of something called arabinose sugar, encodes for a protein that will then make what's called green fluorescence protein, and it will actually glow under UV light. So right now, what you should be thinking about is, what do I expect to see on all of these plates? And then, what do I expect to see on all these plates if I put them under UV light? Now that our 10 minutes has passed, what we're going to do is we're going to put our 2 tubes that have been sitting on ice into this foam holder. And I'm going to put both of the tubes in there so that the bottom, the part that actually has the liquid in it, is sticking out. So it's sticking out on the bottom there. Now this is what I'm going to put into my hot water bath. My hot water bath is set at 42 degrees Celsius, and this acts as what we call a heat shock. And the purpose of the heat shock is to actually open up that plasma membrane for a brief second, so that then that plasmid DNA that I've kind of mixed into my solution can move into the cells, into the bacterial cells. So I'm going to take these, I'm going to come over here to my water bath, and I'm going to put them in the water for 50 seconds. Once our 50 seconds has passed, you'll see in the directions it says you need to get it back onto the ice immediately. This is very important because we kind of want to close up those plasma membranes again and make sure that that plasmid DNA that we want in our bacterial cells kind of get stuck into those bacterial cells. We don't expect that every single bacteria cell that's in there will actually get the plasmid DNA in it, but hopefully enough of them will that we'll be able to see that in our results. So we're keeping this on ice for 2 more minutes, and then what we're doing is we're adding some of the LB broth into my tubes. So the LB broth, this is similar to what our plates are made out of, and our procedure says to add 250 microliters to each of our tubes. So I've added the 250 microliters to my plus pGLO, and I'm going to do the same thing with my minus pGLO. So I'm adding the LB broth, and then I'm going to be incubating it for 10 more minutes. Now I don't need it on ice anymore, so I'm actually going to take it off, and I'm going to let it incubate for those last 10 minutes at room temperature. Once our 10 minutes has passed, I'm going to now flick these around a little bit just to make sure that everything is mixed the way that it should be. And I'm actually going to add these to my plates now. If you look at your procedure, there's a couple ways you could do this. It has asked us to just kind of measure out so that we're adding the same amount to each of our plates, which is the safest way to do it. So we're going to use a micropipette to add 100 microliters to each of our plates. Now here, the most important thing is that you're adding the minus pGLO to the plates that you labeled minus pGLO, and the plus pGLO to the ones that you labeled plus pGLO. And the other most important thing is making sure that you have a clean micropipette tip each time you do this. You do not want to kind of mix your samples. So my orange tube is the minus pGLO. So to help myself, I'm just going to flip over both the ones that I'm going to use, that I know I'm going to use, so I don't mess it up. And I'm just going to add this right into the middle of my plate, and I will move it around later. I will streak my plate at the end. So I'm going to add my sample for both of them. And I don't really want my bacteria to grow in this like pile, so I need to kind of move it around. And again, I'm going to need a clean, sterile loop for each of my samples. You don't want any cross-contamination here. And when I pick up my plate, I don't need to be super precise. I just want to make sure that I get bacteria over my entire plate. So really, when you do this, you're just going to go back and forth, making sure that you're spreading out your bacteria. So now I've done both of my samples that have the minus pGLO, so my regular bacteria samples. And I'm going to go ahead and do the same exact thing with my samples that have the pGLO in them. And if my transformation has worked properly, these bacteria cells, or some of these bacteria cells, should have that plasmid incorporated into their cells. I'm going to do the same thing. We're almost there. Once I get my plates completely spread, I will then have to transfer them to an incubator, which is going to be set at 37 degrees Celsius. And I will actually leave them overnight to allow the bacteria to grow. My hope is that when I come in the next morning, that I will be able to see some bacterial or a lot of bacterial growth on my regular plate, the LB plate with nothing added to it, and maybe some different results on my other plate based on the hypotheses that we've made. So what I would do is I'd add my lab group name to these, right, if there were lots of samples, and I would actually flip them upside down. This is to prevent any condensation that might build on the lid from dropping onto my plate and messing up my results. So I would kind of stack them all like this, upside down, put them in my incubator at 37 degrees Celsius for overnight, and then come in the next day to look at our results. So I'm gonna go put these in my incubator. I'm gonna grab the ones that I put in last night, and we'll take a look at them. All right, so now I've grabbed all 4 of my plates from my incubator that I left overnight, and right off the bat, these are some results that I expected to see. I don't know for sure yet if everything has worked, but here's what I can tell. My regular LB plate that got the bacteria without the pGLO on it grew lots of E. coli. My plate that got the regular bacteria without the plasmid but had ampicillin on it appears to have no bacterial colonies. My other 2 plates that both have the pGLO plasmid in them appear to have some bacterial colonies, but not as much as the original LB plate without the ampicillin. But what I do know is when I compare this plate that has ampicillin and this plate that has ampicillin, there's a clear difference. One of them has some bacterial growth, the other one has none. Just at glance from what I see right here, there doesn't appear to be much of a difference between these 2 plates, my 2 plates that have the pGLO plasmid on them. But I know that this plate right here has an extra thing in the LB broth. It has arabinose sugar. And that arabinose sugar is what we expect to allow the creation of a protein called green fluorescent protein that if put under UV light, we would expect to see some glowing, kind of like what you would see in a jellyfish. So in order to really test if my results are accurate, I need to put all of these under UV light because I want to make sure that the ones that I don't think should be glowing aren't glowing, and the one that I do think should be glowing does. So I'm going to grab my UV light box and we're going to test all 4 samples. The first plate we're going to look at is our regular plate, the LB broth with just the regular bacteria on it. So of course I can see just with my eyes that there's a lot of bacteria on there, but I just want to confirm that it doesn't actually glow, because if all of them are glowing, then I'm not really proving the fact that the arabinose sugar is what is helping to make that protein. And when I turn my UV light on, I can tell there's really not much of a difference. I'm not seeing any type of glowing happening here. Just to make sure there's no like hidden glowing things in this plate, even though I can tell there's really no bacteria on it, it's always safe to check. What you do for one sample, you definitely want to do for all of them. Here I can tell my plate is still completely empty. So this is my plate that has the ampicillin on it, but the negative pGLO. There's nothing added to my bacteria. The third one I'm going to check is the one that has the ampicillin on the plate and the pGLO plasmid. So again, I have some bacterial growth, but it seems to be just regular bacterial growth. There's no glowing happening here under the UV light. My final one that I want to check is the one that has the arabinose sugar. So if all goes well, when I turn on the UV light for this one, I should expect to see not only these bacterial colonies that I see here growing even on the ampicillin, but also I should expect to see some glowing from the creation of that green fluorescence protein. And there we have it. So on this plate, what we know is I do have ampicillin, I do have arabinose, but my bacteria has picked up that plasmid. And so it is now expressing two different traits. It's expressing a ability to still grow on the ampicillin, so something that's making it ampicillin resistant, and it's expressing a gene that encodes for a protein called green fluorescent protein, which is allowing it to actually glow under this UV light. So now we have all 4 of our plates. We got the results we were looking for, which is always nice, not always the case. And you can use this information to answer the rest of our analysis questions.
You have to say thanks. Bye.